Good morning, everyone. Welcome to today's NEXTalk Webinar, From One Cell to Thousands of Insights, Gene Expression at Single-Cell Resolution. Thank you all for joining us. I'm Meng T, and I'm the moderator for today. To make sure everyone has a great experience, here are the few things to know. These sessions will run for about 30 minutes with a Q&A session at the end. If you have any questions during the webinar, please feel free to submit them through the chat box, and we will address them after the presentation. Lastly, please keep your microphone muted throughout the session. In today's webinar, we are happy to have Mr. Chi Hong as our speaker. As the Field Application Manager at NextGene Scientific, Chi Hong has experience for over a decade in his field. Today, he's going to walk us through the world of 10x genomics and show us how single-cell technologies is advancing the gene expression research. Without any further ado, let's start the webinar today. Chi Hong, the floor is yours now.
Okay, hi everyone, good morning. Okay, so welcome back from the holiday. Okay, so I would like to wish all Muslim friends a belated happy Selamat Hari Raya, and also welcome back to the daily life. Okay, so today is our session of webinar with the title From One Cell to Thousands of Insights, Gene Expression Level at Single-Cell Resolution. I'm Chi Hong. Okay, so I'm from NextGene. We NextGene are the distributor of 10x Genomics, so which is actually also a service provider, a solution provider for single-cell sequencing. Okay, without further ado, let's look at some of the agenda for today's sharing session. Okay, so first of all, I will actually give a short and brief introduction towards what is single-cell sequencing. And next up, we actually look at how 10x genomic single-cell sequencing works. Next, we will see what could single-cell sequencing offers compared to other technology or techniques, and how can this offer actually impact our research. And lastly, we can look at what other things is in addition to gene expression that single-cell sequencing can offer in terms of multi-omic research. Okay, after the sharing session, I will actually take a short session of showing you how the result of single-cell sequencing being shown on the software. Okay, single-cell sequencing gene expression. So what we actually learned from the biology class, we all know that DNA is actually the code and then storage for all genetic material for all kind of organism. Okay, so whenever this information is needed to be used, the DNA's information will be expressed into messenger RNA, and then this information will then be translated into protein. So protein is actually the functional unit of the carry out the cellular function. Okay, and therefore the information is actually stored in DNA. Okay. However, when we actually look at ourselves, we realize that although the DNA content for all cells of a single individual are supposed to be the same, the phenotype or the appearance of this cell are actually very different, and also what kind of function it can carry out also very, very different. For example, we have skin cell that is actually very different from our eyes, the eyeball cell. Okay, one we can actually use to see things, another we can only touch things to get information. Okay, this is weird because DNA are supposed to be the same, and therefore the explanation for that is that they are different RNA being expressed to enable different function and also to carry out the task that is actually need to be, designed to be working. Okay, so therefore, even though the DNA content is the same, the RNA content of individual cell of a single being are actually different. So this RNA profiling is the key to cellular phenotyping. So it is the key of the RNA content which actually decide what can the cell do. So there are several technology or techniques that allow us to study the RNA profiling of different cells. Okay, so if we are just targeting several genes, so the most best technique to study will be using the reverse transcription qPCR method to target just a few genes. However, when we actually want to know the near whole transcriptome sequencing, means that sequence all genes within a different cell. So the method of choice will be either microarray or the conventional RNA sequencing using the NGS or next generation sequencing technology. So here we actually refer it to be bulk RNA sequencing. So these two techniques, the microarray and also the RNA sequencing, are able to sequence the entire transcriptome or all transcript being expressed inside cells. Okay, so the advantage of RNA sequencing compared to microarray is that RNA sequencing are able to offer information of novel, means that not previously discovered, new gene, new transcript. However, both of them share one same feature, which is actually they only average out the gene expression, meaning that they treat all cells equally. So let's say, using an example of a tissue sample, so the tissue of a human being is supposed to contain epithelial cell, endothelial cell, fibroblast, and even muscle cell and all that. Okay. When we're using microarray or RNA, bulk RNA sequencing technology, the RNA content of all these cells will be actually mixed together. And then the different RNA expression, supposedly on different gene, they will be actually mixed and then even out. So therefore, we don't see gene expression for a particular cell type, but the average of all cell type for, let's say, a tissue sample. So if you are interested to know that the gene expression RNA profile for different cell type, okay, so we have this option, which is actually the single-cell RNA sequencing. So this sequencing technology is able to provide expression profile on individual cell, and then by using this expression profile on individual cell, we've grouped them into cell cluster and get information of the RNA content for different cell type. Here, I would like to go a little bit insight about the different technology that used to study, let's say, RNA or protein. So for flow cytometry, is actually a technique to study the protein or cell surface marker that actually express on the surface of cell. So it's able to measure multiple marker at once. It's able to offer high throughput analysis of thousands of cells per second. It's actually quite low cost per experiment, although we need to maintain the flow cytometry machine. For microarray technology, is able to quantify the expression level of specific genes of interest. It examines the expression of 1,000 genes simultaneously. It's a good validation tool and cost-effective for larger studies. For bulk RNA sequencing that using NGS technology, it's actually able to provide information of the whole transcriptome profiling, which is actually quite similar to microarray, but is capable of detecting new gene, new variants, new transcript. Okay, but as mentioned earlier, it's only providing average gene expression for all the cell type that actually mix together. It has a simple workflow and analysis, and it has a low cost and sequencing depth with a focus on population level insights. Okay. Then next on, we have the single-cell RNA sequencing. Again, it's actually able to look at the whole transcriptome profiling, the RNA content of the cell, however, at a single-cell resolution. So we look at the cell on their own, individually, one by one, rather than mixing all the RNA content of different cells together and average out the gene expression data. So how does it help in terms of the data is that you're able to let us capture rare cell type and then also some dynamic state, which means that we have activated cell and also the non-activated cell, we're able to separate them, okay, because the gene expression content of these two states are actually different. So we will then be able to discover new genes, new cell type, or rare population. Okay, So here, since the quite similar in terms of gene expression profiling for conventional bulk RNA sequencing, so I like to further showing you what exactly do I mean by average out the gene expression for bulk RNA sequencing and also the gene expression profile for each of the cell type under single cell sequencing. Okay, so this one actually, this is gene A, gene B, gene C, gene D, gene E, and F. Okay, so this gene we actually look at the one single expression level, even though they are different cell type, because the RNA expression level are being mixed and then being shown as an individual single gene. Whereas in single cell sequencing, because we actually sequence the cell individually, so we have, let's say, cell type A, so we look at the gene's expression level for this cell type only, and then in the same sample, if you have cell type B, we can be able to look at the inside of this cell, the gene expression profile, so on for cells type C, cell type D, and so on and so forth. Okay, so depending on how many different cell type within your sample, we were able to separate the gene expression profile for this different cell type. Whereas in the conventional RNA sequencing, it will be all averaged out to show as one expression level. Okay. So using an analogy that normally used in 10x genomics, bulk RNA sequencing is like a smoothie that actually blend all the different fruit together, and when we taste it, we get a hint or sense or taste of one of the fruit. Okay, so we suspect that there are some strawberry inside, but we can't know for sure. But with the help of the single cell RNA sequencing, we are able to separate the fruit type, okay, and then we actually look at them individually and then compare them among each other. So single cell sequencing technology actually is not something that very new. It actually already available since 10 to 20 years ago. However, at the time, it's actually a technique that is considered homebrew, and therefore the protocol, the workflow is actually not very established and considered very challenging. So at that time, only a handful number of cells can be sequenced. For example, like 85 cell, 37 cell, or 34 cell. So with the offering from commercial company, now we have established protocol that make the entire workflow more streamlined and more established. Now we can easily sequence 1,080,000 cell, 49,000 cell, or like 200,000 cell without any issue. So this is an example of gene expression data from single cell. Okay, so after we actually get the RNA profile of a single cell, we will actually group them based on the similarity of the RNA profile. So if these two individual cells have similar RNA profile, most likely they are the same cell type, and therefore we group them under the same cluster. Okay, and then if they are another distinct RNA profile and clustering, we have another cell type. Okay, so on and so forth. So over here is a real presentation of the data. So this particular circle here are circling many cells. Actually, we actually identify it as myeloid cell. And then this small cluster of cell is actually epithelial cell. Over here, plasma, natural killer cell, and this bunch of cells are actually B cell. Okay, so they are clustered and grouped based on the similarity of the RNA profile of this individual cell that we sequence. Okay, so after the introduction on the single cell, now we take a look at how actually we're able to get the data itself. Okay. So first of all, of course, we need a sample to give us the information. So single cell sequencing technology is actually species agnostic. So we can actually use sample like mammals from human, monkey, rats, okay? And then we can also use like from species like fish, and then plant, and even insects. So any species is actually possible. Secondly, the cell type, the sample type. Okay, so we need to use a cell suspension. So if let's say we are using a tissue sample, we will actually need to disrupt and dissociate this tissue sample into single individual cell, okay, suspension. And then for sample that like blood cell, because they are single cell in nature, so we do not need further processing of this. Okay, then this is actually the type of sample that we actually to begin with. So if necessary, this tissue can be frozen. It could be fixed using PFA, OCT, or even fixed into formalin fixed paraffin embedded sample. Okay. For if let's say there's a need to collect more sample, so while waiting, then we fix the cell first, or we store it frozen first. After getting the sample ready, so with a single cell suspension, we can then using the reagents from 10x Genomics. So let's say the gel beads, the library preparation construction reagent. Okay, so we actually prepare the cell, and then after that make it into a sequenceable library, and then we actually perform NGS to sequence those library. Okay, once the sequencing data is being produced, we'll input it into a free software offered by 10x Genomics to go through the analysis pipeline and visualize the result in the example earlier. Okay, a little bit in-depth explanation on how we actually able to get the data. Okay, so here is actually showing a reagent from 10x Genomics that we call gel beads. It's actually a round shaped beads. Then on the surface of it, we have some unique sequence, okay, unique barcode sequence. Okay. So in this reagent, there are many different, more than millions of gel beads. Each of them have different barcode sequences. Okay. So in the microfluidic chip that we loaded the barcode as well as the cell. Okay. So one single cell will actually meet with one unique sequence barcode. And then there will be engulfed and surrounded by an oil layer to separate from each other. So this oil formation that surrounding individual cell and also a single unique sequence gel bead, we call it the gel bead emulsion. Okay, and then of course the cell will be lysed, and the RNA content of this single cell will acquire the unique barcode. Okay, so therefore individual cell will acquire unique barcode and being sequenced, and all the sequences that has the same unique barcode are actually derived from the same cell. So currently we are able to recover more than 100 and up to 20,000 of cell for an individual sample, up to 8 sample, and capture it up to 80%. Okay. Here is showing the microfluidic chip that we loaded the sample, the gel beads, and this is actually the suspension of the gel bead emulsion, where the single cell is actually meet up with the single cell, and then after that surrounded by the oil. Okay. So next, we will take a look at what could 10x Genomics single cell offer to your research. So a brief comparison of what kind of information we might get from single cell sequencing is, let's say, using this study of tissue development by Jowick as example. So bulk RNA sequencing needs the unique version of gene and short life in between cell state. So by using single cell sequencing, they are able to find rare state and special gene patterns from their sample, and this helps the scientists understand how healthy tissue grows and how diseases started. So in another study by Pack, they actually study on heart development. So they realized that bulk RNA sequencing averaged out all heart cells, and therefore hiding a rare cell type that actually they call it like pacemaker. which is actually important for heartbeat control. So by using single-cell sequencing, they're able to reveal this rare cell type, the pacemaker, and track how they develop. So this could be very useful to help to treat heart rhythm problems. And in terms of drug safety testing, so bulk RNA sequence, they consider all cells are actually the same, they react the same to the drug testing. So they might be missing out a small subset of group that are actually harmful or susceptible to the drugs. So by using single-cell sequencing, the publication actually spotted a 3% cell or group that's showing early warning of inflammation. So this could further improve unsafe drugs from advancing and save some developmental costs. And last but not least, the example here actually is a study using cancer treatment monitoring. So bulk RNA sequencing blur out immune cell changes during therapy, making it look like nothing had shifted. Again, with single-cell sequencing, we're able to pick up an increase in killer T cell and a drop in suppressor T cell. This is actually an early sign that the treatment was working. Okay? So this allowed the clinician to better understand the result from the cancer treatment monitor. So overall, what single-cell sequencing can offer is actually the characterization and identification of heterogeneous cell population. Okay, so the key feature of single-cell RNA sequencing data is that we are able to sequence individual cells, group cells with similar RNA profile into a cell clustering. Okay, so this cell cluster is supposed to be the same cell. That is actually a same cell type, for example, a T helper naive cell, the same cell type, and then further distinct into T helper memory cell, T effector cell, B cell, monocyte, and all that. Okay, so therefore, with this data, we're able to analyze cell population, okay, because we actually separate them into different cell types, and enable to identify some challenging cell type like neutrophil or even some small population or rare population. So after getting the cell cluster, we can then compare the gene expression profile between the cluster, means that the different cell type. We can actually compare the cell cluster, which actually potentially different cell state. For example, we're able to separate T cell with different RNA profile because when they're activated and resting, they express different genes. Okay, so we can compare this cell, T cell, with different cell state. Also, we can actually compare cells population between different conditions. So having that information that genes that are differentially expressed, we can then perform, of course, functional enrichment analysis to better understanding of the biological meaning, for example, the pathway or processes. Okay, so this is an overview of the presentation of the data, okay, after a single-cell sequencing data output analysis. Okay, I will actually further show you guys on this later on using a software. But overall, this is actually different cell cluster, different cell type with a unique expression profile. We can select individual cluster, and then we can compare different cluster. Okay, next I would like to share some insights on how single-cell sequencing actually impact your research by using some publication. Okay, so this will be the summary of those applications that I'm going to share with you later. So basically, the unique feature of single-cell sequencing will be able to characterize and identify heterogeneous cell population. So with this information, we are able to uncover novel cell type novel cell state or rare cell type. So after that, we are able to discover new cell marker by using the gene expression data and their regulatory pathway. By using the same information, we are able to see the development of the cell, and then after that to reveal the hierarchy or lineage relationship. Also, we can actually profile healthy and diseased tissue and then compare them to uncover again the new cell marker and also regulatory pathway. Okay. So in this particular publication by Lin, okay, so this will be actually showing of the cell cluster from the data generated. So we're able to see that the cell type or cell state present in the tissue sample that they use. So like memory cell, natural killer cell, naive B cell. Okay, we can also get like the proportion of different cell type, like the number of cells. So when there are more population number, the bigger cluster means that there are more cells. Okay, then how does the gene expression program? So we can select the cell cluster and then compare to another cluster to see what genes are actually upregulated and downregulated. So also we can also identify rare cell type. So one of the key findings from this particular paper by Lin, actually they discover a CD8 plus NK cell, the unique cell cluster in nasopharyngeal cancer tumor tissue. Okay, so this CD8 plus natural killer cell are actually distinct cell population here, and normally not found in normal nasopharyngeal tissue. So previously, this particular cell type has only reported in other diseases associated with HIV and EBV, but never been previously reported in nasopharyngeal cancer. Okay, so in another publication, the author actually comparing different cell tissue type, okay, the disease and also the normal, and then they compare the cluster of cell, the gene expression profile from all these different cell type. So thereby they're actually able to identify a unique cell cluster 17 that actually has a signature PAM50 gene. So this gene actually signature the cluster to be the cancerous cell cluster, okay, based on the comparison of different tissue types. So after having the cell cluster, which is actually different cell type, we can actually study like the interaction between these different cell type. How does this interaction affect the gene expression level as well? So in this particular publication by Liu, okay, they actually realized and found that the T cell that actually infiltrating the tumor site, okay, so they actually become suppressive by the tumor cell. Okay, so the tumor cell actually regulate the T cell to make them actually non-aggressive and submissive and not actually attacking the tumor cell. Okay, so this is a key finding of them that using the single-cell sequencing, comparing the expression profile of the T cell actually that meet up with the tumor cell. So next up, this is actually a publication using plant, a Texas leaf. Okay, so they're actually looking for the regulation of bioactive ingredient biosynthesis in the leaf. Okay, so they are in this study they actually characterize and the cell and group it into 15 cluster. So 15 cell type, which is actually considered quite high for the leaf. So the finding is that they found that the taxol biosynthesis gene are present mainly in the leaf mesophyll cells. So whereas for phenolic acid and flavonoid biosynthesis gene, they are actually in different cell type, which is actually the leaf epidermal cell, and therefore the terpenoid and also steroid biosynthesis genes are mostly expressed in the leaf mesophyll cell as well. Okay, by using the single-cell sequencing data, and also the cell clustering comparison, we're able to identify the different gene expression profile and the biosynthesis pathway that highly present in a certain cell type. Okay, so next up, and also a publication on tomato. Okay, so they're actually able to identify a functional tomato transcription factor, which is actually the MYB92 and also the ASFT enzyme. So these two transcription factors and also the enzyme demonstrate that it is actually important for the exodermal suberin during the drought resistant time. So they're able to produce this suberin for as a plant water deficit response. Okay, so the last publication that I would like to share over here will be the study on lineage-specific development using the single cell. So the precursor cell is actually the retinal progenitor cell, and how they actually produce different lineage like the neurogenic lineage. So the PR precursor cell, HC precursor cell, and cluster B cell. Okay, so by using this sequencing technology, they are able to cluster them, all the cell type in the tissue, and then we identify the progression or the development from the RPC progenitor cell into the later lineage of for the development of the eyes in zebrafish embryo. Okay, so previously we are actually looking at all the information using the gene expression data, but actually the 10x genomics single-cell sequencing also capable of offering other information. For example, the immune profiling to offer the sequence of the T cell receptor or B cell receptor as well. And then we can actually include cell surface protein identification using antibody to bind the surface protein using antibody and identify the presence of these markers on individual cell. We can also look at the ATAC-seq to look at the condition of the whether the chromatin is actually open up for the transcription to be activated, and then correlate it with the gene expression data. Okay. So as a summary of today's sharing, where can single-cell sequencing take us? One of the key features is actually the complete characterization of different tissue type. And we understand the interaction of these different cell type. And then through it, we're able to understand the regulatory pathway and discover a biomarker. So here I would like to show you the analysis workflow that actually including the cell subpopulation identification through the use of grouping cell with similar RNA content into a same cluster. So the analysis software from 10x Genomics, which is actually the analysis pipeline of Cell Ranger, and the viewing of the result, the root browser, are actually a free software. Okay, so here I would like to change to the software to give you a brief overview of what kind of result can be you guys can getting from the data. So this is actually the loop browser actually showing you the result of a sequencing data. So over here you can see there are different clusters. So let's say cluster 1, cluster 2, cluster 3, so on and so forth. This cluster actually are cells that actually having the same RNA expression profile. Okay, so if you are interested in certain cell cluster and you want to compare them, you can actually highlight them, and then over here you have the run differential expression tab. Okay, you can actually just compare the expression of genes for these two clusters. Okay, so for example, okay, So let's say over here, I'm comparing the two cluster. This one I highlighted as test. Okay, and then this one I highlighted as something new. Okay, so I select individual cell cluster that I'm interested, and then I put it over here, and then I'm comparing them. So here we'll be showing, let's say, the genes that are differentially expressed. Yeah, I hope that you guys can see it without issue, the words over here. And then if there are any genes that you are very interested in particular, you can actually go to the feature tabs, and then you type the gene name. So let's say, KRT. Okay, so you can actually select the type of gene, okay, and then see which cell actually have highly expressed of certain genes. Okay, so okay, and then you can actually compare the, you can select them and add it into as a new cluster, and then you can actually perform the analysis to look at the gene expression, and then also you can actually get a Wiley plot. This one is actually only for CD474 cell. Okay, and yeah. So you can actually change the view of it as well. So, okay, can actually change it into, let's say, a UMAP. Okay, and then you can split all this cluster into individual, let's say, cluster 1, cluster 2. This is actually how you're actually able to select, let's say, the cluster that you're interested, or further select the cluster that you're interested. Okay, you can actually, let's say, if you're interested on this particular 2 cell, okay, I think 2 cells is too little. Say, is there any bigger cell? Let's say this one. Okay. So let's say you're interested on this group of cell, you can actually use a lasso tool, and then you actually select it. Okay, then you can actually save this particular cell cluster as a new cluster. Let's say new. Okay, and then you can actually add into certain group so that you can perform the analysis of all the cell type under the same group. Okay, let's say a new cluster. Okay. So now you have 3 cell cluster that can actually perform, let's say, the gene expression comparison or the expression of the gene. Okay. If let's say you feel like the individual genes selection is actually a bit too handy, you can actually compile a list of gene, and then after that you upload it, and you actually open up, and then you actually select those cells with the gene that you're interested as well. Okay, so I think I don't want to make it too lengthy, so I will stop here about the showing of the cell cluster. Okay, of course, you can actually name the cell cluster to make it into a cell type. Let's say T cell or whatever. Okay, so yeah, so I think I will actually stop here for the demonstration of the brief demonstration of the software. I think we can move on to the Q&A session.
Yeah, thank you, Chi Hong, for the insightful presentation on the gene expression at single-cell resolution. So now we will move into the Q&A session. You can unmute yourself to ask a question, or you can type in the chat box. So Chi Hong will address accordingly. Is there any question from the floor?
Hello.
Hello, yes.
Hello. Sorry, I'm very new to this, so I just want to understand the results, right? So you mentioned that we can discover new types of cells, right? Unique cells using this analysis.
Yes.
So from the results that you showed just now, the dots and everything, how do we know like this is a new type or unique cell or unique cell population?
Okay, that will actually based on some unique genes markers. So of course, we need to actually analyze the data, and then after that found some like a new gene that actually never found before in other cell type. Okay, and then yeah, so at first then there will be further analysis to identify it as a new cell type. Okay, so because let's say in the example of the one of the publication earlier, okay, so one more. Okay, so so in this particular example, it's actually showing you a cell type with a signature of a CD8 expression NK cell. So first we need to identify, let's say, the unique gene marker for NK cell, and then after that we look for other markers like CD8. Okay, then because we have, let's say over here, NK cell with CD8 and NK cell without CD8. Okay, so first we need to identify the cell type, and after that we look for the why it actually separate into two groups. So to look for, let's say, the different gene marker that express for these two clusters. Or let's say normally we have the understanding of the sample that you are studying on, and then you actually, let's say, go through the publication on those literature review. So you get, let's say, the normal cell type are being found over there. So after you actually characterize those cell type, then the new one, then we actually need to look at the unique gene marker for those cluster and see what could be potentially is the new cell type.
Okay, understood. Thank you.
Thank you.
Okay, thank you. We have another question from the chat box from Chai Yi Quan. Is anyone has using this technology in Malaysia?
Yeah. We have a few groups, namely in University Malaya, and then we have like in hospital children in Cheras, and then we have institute like IMR and all that are currently using the 10x genomic single-cell sequencing technology.
And another question from Mar Hikmah. What is the lowest starting material that can be used?
Lowest, if I'm not sure, do you mean like the lowest cell number? If you're talking about the minimum cell number, currently we can sequence a minimum of 100 cell. Okay, because below that, then we are a bit hard to differentiate whether it's really a cell or some background RNA content. So yeah, if you're talking about the lowest number of cell we can start with, then it's actually, we recommend to be 100 and ideally 500 cells.
Okay, another question from G Brew. What is the name of the software?
Okay, so for this one, I think I better show this slide again. Okay, okay, it's actually, but however, these two software is actually currently used only for 10x genomics data. So the name of the software for the analysis pipeline, okay, so the workflow is actually Cell Ranger. So it will generate an output file. So we loaded the output file using another software to view the cell cluster and to do some manipulation. That one will be actually the loop browser.
Okay, do we have any more questions from the floor? Well, I think it's well concluded. So thank you so much for Chi Hong for the informative presentations today. And a big thank you to you all to participate in this session. We hope you find the information shared is valuable and inspire you to apply in your own research. And we would like to hear your thoughts for today's session. If you have a quick moment, please fill out the survey in the chat and we will be sending the certificate to you once you have completed the form. Lastly, if you have any question, please feel free to reach us out at sales@nextgene.ny. And thank you again, and we will see you in our next future events. Thank you. Bye bye.
Thank you, everyone. Bye bye.